Hello and welcome dear students. Today we are going to discuss about the topic pedigree analysis and expressivity. The main objectives of today's lecture are to know the basics of pedigree analysis, to discuss about the different modes of inheritance, to discuss about the sporadic and non-heritable diseases, to understand the characteristic predictions through pedigree analysis, and finally to discuss about multifactorial traits using the pedigree as a tool to collect a family history of common chronic disorders. Dear students, let's start with the introduction first. Gregor John Mendel worked on the principles of inheritance in genetics a long time ago. His work helped to understand the fundamentals of genetics and inheritance patterns in living organisms. Later scientists developed another approach to help us understand the inheritance of genes in living organisms from parents to offsprings. This approach is known as the pedigree analysis. Pedigree analysis was developed to understand the inheritance of genes from parents to the offsprings. It was developed as a chart that can represent a family tree along with the family members and their genetic traits respectively. Gregor John Mendel's experiments showed that the factors that we know as genes are the factors which are responsible for the inheritance of traits or characters from parents to the offspring. Therefore, genes are the hereditary unit of organisms which are responsible for carrying the information from parents to their offsprings. These genes are actually responsible for the characteristics of a living organism and can also be the reason for some disorders present in them. Mendelian disorders are genetic diseases which follow a Mendelian inheritance pattern, and their inheritance is controlled by Mendelian genetics. Therefore, by studying the family history of an individual through pedigree analysis, we can predict whether an individual is at risk of a genetic disorder or not. Pedigree analysis is a scientific approach to study the inheritance of human genes. So, pedigree analysis can be used to easily understand family history and the inheritance of dominant and recessive traits. With the help of a pedigree chart, one can easily identify dominant and recessive and also the carrier of these traits. A pedigree chart is a diagrammatic representation, that is chart of a family where one can easily determine the occurrence of a phenotype in an organism genes passed down from their ancestors. A pedigree is actually a diagram showing the ancestral relationship and transmission of genetic traits over several generations in a family. Square symbols are almost always used to represent males, while circles are used for females. Pedigrees are used to help detect many different genetic diseases. A pedigree can also be used to help determine the chances for a parent to produce an offspring with a specific trait. 4 different traits can be identified by pedigree chart analysis, like autosomal dominant, autosomal recessive, X-linked or Y-linked. Inbreeding or mating between closely related organisms can clearly be seen on pedigree chart. Genetic counselors commonly use pedigrees to help couples determine if the parents will be able to produce healthy children or with any disorder. A pedigree results in the presentation of a family information in the form of an easily readable chart. Pedigree construction is a family history, and details about an earlier generation may be uncertain as memories fade. Dear students, the relationship in a pedigrees are shown as a series of lines. Parents are connected with each other by a horizontal line, and a vertical line leads to their offsprings. The offsprings are connected by a horizontal sibship line and listed in birth order from left to right. If the offspring are twins, then they will be connected by a triangle. If an offspring dies, then its symbol will be crossed by a line. If the offspring is still born or aborted, it is represented by a small triangle. Each generation is identified by a Roman numeral 1, 2, 3, and so on. And each individual within the same generation is identified by an Arabic numeral, that's 1, 2, 3, and so on. So analysis of the pedigree using the principles of Mendelian inheritance can determine whether a trait has a dominant or a recessive parent of inheritance. Pedigrees are interesting because they can be used to do some detective work and are often used to study the genetics of inherited diseases as well. For example, pedigrees can be analyzed to determine the mode of transmission for a genetic disease. Dominance, whether the disease alleles are dominant or recessive. Linkage, whether the disease alleles are X-linked on the X chromosome or autosomal. To study the inheritance patterns of genes in humans and other species for which controlled matings are not possible, geneticists use the analysis of pedigrees and populations. Dear student, let's now understand the pedigree analysis in detail. First, the pedigree charts. Pedigree charts are diagrams that show the phenotypes and or genotypes for a particular organism, its ancestors and descendants. While commonly used in human families to track genetic diseases, that can be used for any special and any inherited trait. Geneticists use a standardized set of symbols to represent an individual sex-family relationship and phenotype. These diagrams are used to determine the mode of inheritance of a particular disease or a trait or character and to predict the probability of its appearance among the offsprings. Pedigree analysis is therefore an important tool in basic research, agriculture, and genetic counseling. Each pedigree chart represents all of the available information about the inheritance of a single trait, most often a disease, within a family. The pedigree chart is therefore drawn using factual information, but there is always some possibility of errors in this information, especially when relying on family members, recollections, or even clinical diagnosis. Second, the pedigree chart convention symbols. In pedigree analysis, standardized human pedigree nomenclature is used. If possible, the male partner should be left of female partner on relationship line. Siblings should be listed from left to right in birth order, oldest to youngest. Second, modes of inheritance. Given a pedigree of an uncharacterized disease or trait or character, one of the first tasks is to determine which modes of inheritance are possible and then which mode of inheritance is most likely. This information is essential in calculating the probability that the trait will be inherited in any future offspring. Mostly there are 5 major types of inheritance: autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and Y-linked. Autosomal dominant. When a disease is caused by a dominant allele of a gene, every person with that allele will show symptoms of the disease, and only one disease allele needs to be inherited from an individual to be affected. Thus, every affected individual must have an affected parent. A pedigree with affected individuals is every generation is typical of AD, that's autosomal dominant disease. However, beware that other modes of inheritance can also show the disease in every generation. It's also possible for an affected individual with an autosomal dominant disease to have a family without any affected children if the affected parent is a heterozygote. This is particularly true in small families where the probability of every child inheriting the normal rather than the disease allele is not extremely small. Autosomal dominant diseases are usually rare in populations, therefore, affected individuals with autosomal dominant diseases tend to be heterozygotes. Otherwise, both parents would have had to be affected with the same rare disease. Achondroplastic dwarfism and polydactyly are both examples of human conditions that may follow an autosomal dominant mode of inheritance. Achondroplasia is a common form of dwarfism. FGFR3 gene at 4p16, that is chromosome 4, p-arm region 1 band 6 encodes a receptor protein that negatively regulates bone development. A specific base pair substitution in the gene makes an overactive protein, and this results in shortened bones. So achondroplasia is considered autosomal dominant because the defective proteins made in heterozygous, that is capital A small a embryos, halt bone growth prematurely, and while as in capital ones or the homozygous, that is capital A embryos do not make enough limb bones to survive. Most but not all dominant mutations are also recessive lethal. In achondroplasia, the capital A allele shows dominant visible phenotype, that is shortness, and recessive lethal phenotype. Dear students, let's discuss now the second one, that's X-linked dominant. In X-linked dominant inheritance, the gene responsible for the disease is located on the X chromosome, and the allele that causes the disease is dominant to the normal allele in females. Because females have twice as many X chromosomes as males, females tend to be more frequently affected than males in the population. However, not all pedigrees provide sufficient information to distinguish XD, and that's X-linked dominant and autosomal dominant. One definitive indication that a trait is inherited as autosomal dominant and not X-linked dominant is that an affected father passes the disease to a son. This type of transmission is not possible with X-linked dominant since males inherit their X chromosome from their mothers. Example: Fragile X syndrome, the fMRI gene at XQ21, that is X chromosome, Q-arm, region 2, band 1 encodes a protein needed for neuron development. There is a CGG in repeat array in the 5UT, or that is untranslated region. If there is expansion of the repeat in the germline cell, the child will inherit a non-functional allele. XA/Y, males have fragile X mental retardation, that's IQ is less than 50, because none of their neurons can make FMR1 proteins. So fragile X syndrome is considered X-linked dominant because only some neurons in XA/X small a females can make FMR1 proteins. Dear students, let's now discuss the third type, that's autosomal recessive disease. Autosomal recessive diseases that are inherited in an autosomal recessive pattern require that both parents of an affected individual carry at least one copy of the disease allele. With autosomal recessive traits or characters, many individuals in a pedigree can be carriers, probably without knowing it. Compared to pedigrees of dominant traits, autosomal recessive pedigrees tend to show fewer affected individuals and are more likely than autosomal dominant or X-linked dominant to skip a generation. Thus, the major feature that distinguishes autosomal recessive from autosomal dominant or X-linked dominant is that, unaffected individuals can have affected offsprings. Attached earlobes is a human condition that may follow an autosomal recessive mode of inheritance pattern. Example: phenylketonuria or PKU. Individuals with phenylketonuria have a mutation in the PAH gene at 12q24, that is chromosome 12, Q-arm region 2 band 4, which encodes an enzyme that breaks down phenylalanine into tyrosine called PAH. Without this enzyme PAH, the accumulation of phenylalanine and other metabolites such as phenylpyruvic acid disrupts brain development typically within a year after birth and can lead to intellectual disability. Fortunately, this condition is both easy to diagnose and can be successfully treated with a low phenylalanine diet. There are over 450 different mutant alleles of the PAH gene, so most people with phenylketonuria are compound heterozygotes. Dear students, let's now understand the X-linked recessive. Because males have only one X chromosome which is coming from their mothers, any male that inherits an X-linked recessive disease allele will be affected by it. Therefore, in X-linked recessive modes of inheritance, males tend to be affected more frequently than females in a population. This is in contrast to autosomal recessive and autosomal dominant, where both sexes tend to be affected equally, and X-linked dominant, in which females are affected more frequently. However, in the small sample size typical of human families, it is usually not possible to accurately determine whether one sex is affected more frequently than the others. On the other hand, pedigrees that can be used to definitely establish that an inheritance pattern is not XR is the presence of an affected daughter from unaffected parents, because she would have had to inherit one X chromosome from her father. He would also have been affected in XR, the X-linked recessive character. Dear students, let's discuss an X-linked recessive example, the hemophilia. An F8 gene at XQ28, that is X chromosome QRM region 2 band 8, encodes blood clotting factor 8C. Without factor 8C, internal and external bleeding cannot be stopped. Back in the 1900s, XA/Y males' average life expectancy was 1.4 years, but in the 2000s, it has increased to 65 years with the advent of recombinant human factor 8C. So hemophilia A is recessive because XA/X females have normal blood coagulation, while XaXa females, which are homozygous, have hemophilia. Dear students, let's discuss the other condition, that's Y-linked. Only males are affected in human Y-linked inheritance and other species with the XY sex determining system. There is only father the son transmission. This is the easiest mode of inheritance to identify, but it is one of the rarest because there are so few genes located only on the Y chromosome. An example of Y-linked inheritance is the hairy ear rim phenotype seen in some Indian families. As expected, this trait is passed on from father to all sons and no daughters. Y chromosome DNA polymorphisms can be used to follow the male lineage in large families or through ancient ancestral lineages. Dear students, let's now discuss about the sporadic and non-heritable diseases. Not all of the characterized human traits and diseases are attributed to mutant alleles at a single gene locus or position. Many diseases that have a heritable component have more complex inheritance patterns due to the involvement of multiple genes and because of the environmental factors. On the other hand, some non-genetic diseases may appear to be heritable because they affect multiple members of the same family, but this is due to the family members being exposed to the same toxins or other environmental factors like in their homes. Finally, diseases with similar symptoms may have different causes, some of which may be genetic while others are not. One example of this is ALS, that is amyotrophic lateral sclerosis. Approximately 5 to 10% of cases are inherited in an autosomal dominant pattern of inheritance, while the majority of the remaining cases appear to be sporadic. In other words, not caused by a mutation inherited from a parent. Different genes or proteins are affected in the inherited and sporadic forms of ALS. The physicist Stephen Hawking and baseball player Lou Gehrig both suffered from sporadic ALS. Dear students, let's now discuss calculating probabilities. Once the mode of inheritance of disease or trait or character is identified, some inferences about the genotype of individuals in a pedigree can be made based on their phenotypes and where they appear in the family tree. Given these genotypes, it is possible to calculate the probability of a particular genotype being inherited in subsequent generations. This can be useful in genetic counseling, for example, when prospective parents wish to know the likelihood of their offspring inheriting a disease for which they have a family history. Probabilities in pedigrees are calculated using knowledge of Mendelian inheritance and the same basic methods as are used in other fields. The first formula is the product rule. The joint probability of two independent events is the product of their individual probabilities. This is the probability of one event and another event occurring. For example, the probability of a rolling a 5 with a single throw of a single 6-sided die is 1/6, and the probability of rolling 5 in each of 3 successive rolls is 1/6 multiplied by 1/6 multiplied by 1/6 is equal to 1 divided by 216. The second useful formula is the sum rule, which states that the combined probability of two independent events is the sum of their individual probabilities. This is the probability of one event or another event occurring. For example, the probability of rolling a 5 or 6 in a single throw of a dice is 1/6 plus 1/6 is equal to 1/3. With these rules in mind, it is possible to calculate the probability that two carriers, that's heterozygotes, of an autosomal recessive disease will have a child affected with the disease as 1/2 multiplied by 1/2. Dear students, let's now discuss the pedigree in human use. A pedigree may be used to establish the probability of a child having a particular disorder or condition. It may be used to discover where the genes in question are located, X, Y, or autosomal chromosome, and to determine whether a trait is dominant or recessive or X-linked or Y-linked. When a pedigree shows a condition appearing in a 50-50 ratio between men and women, it is considered autosomal. When the condition predominantly affects males in the pedigree, it is considered X-linked. Some examples of dominant traits include male baldness, astigmatism, and dwarfism. Some examples of recessive traits include small eyes, little body hair, and tall stature. Dear students, let's now discuss about the characteristic predictions through pedigree analysis. While the pedigree analysis does not help predict characteristics like height, other features such as hair color, blood group, and eye color can be easily found. To understand pedigree analysis, one needs to be clear about dominant and recessive genes. Let's consider an example. The color blindness trait can be found in the X or Y chromosome. However, as this trait is common in the X chromosome, color blindness commonly occurs in males. However, females can also be affected if they inherit the X chromosome with color blindness from the affected mother or father. To understand how characteristics of living things are passed from generations to generation, a family tree or pedigree analysis or a pedigree chart is often used to illustrate the movement of the characteristics through each generation. The pedigree may involve single gene inheritance or the inheritance of multifactorial traits or characters, that is, multiple gene and environmental interaction. Dear students, let's now discuss about multifactorial traits using the pedigree as a tool to collect a family history of common chronic disorders. Unlike single gene disorders that are very rare, common chronic disorders such as heart diseases, cancer, and diabetes may either directly or indirectly impact our lives. These conditions have been found to run in families, and scientists now know that the appearance of these disorders is influenced by our genetics, environment, and behavior. Collecting family history with a pedigree for these chronic disorders is the first step towards prevention. Sometimes assigning genotypes for family members at risk for multiple chronic disorders is not easy. That's because many common chronic conditions are not controlled by single genes but are multifactorial, meaning they are influenced by multiple genes and the environment. Most of our traits fall into this category. There are traits that vary slightly from individual to individual, such as height and hair color, as well as common chronic conditions like heart diseases, diabetes, and cancer. Dear students, let's now conclude the lecture. Pedigree analysis is a scientific approach to study the inheritance of human genes. Pedigree analysis can be used to easily understand family history and the inheritance of dominant and recessive traits. With the help of a pedigree chart, one can easily identify dominant and recessive traits and also the carrier of these traits. Pedigree charts are diagrams that show the phenotypes and or genotypes for a particular organism and its ancestors. While commonly used in human families to track genetic diseases, they can be used for any species and any inherited trait. Geneticists use a standardized set of symbols to represent an individual sex, family relationship, and phenotype. These diagrams are used to determine the mode of inheritance of particular disease or trait or character and to predict the probability of its appearance among the offsprings. Pedigree analysis is therefore an important tool in both basic research and genetic counseling. Each pedigree chart represents all of the available information about the inheritance of a single trait, most often a disease, within a family. The pedigree chart is therefore drawn using factual information, but there is always some possibility of errors in this information, especially when relying on family members' recollection or even clinical diagnosis. A pedigree may be drawn when trying to determine the nature of a newly discovered disease or when an individual with a family history of a disease wants to know the probability of passing the disease on to the children. Pedigree analysis can be used to determine the mode of inheritance of specific traits such as diseases. If the mode of inheritance is known, a pedigree can be used to calculate the probability of inheritance of a particular genotype by an individual. Dear students, it was all about today's lecture regarding pedigree analysis and expressivity. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.